Thank you very much, Prof. Thank you very much. My name is Ralph Titemlalo, and I'm the Managing Director for HG&H Pharmaceuticals. I'm a pharmacist by profession. Studied at Rhodes University, where I also obtained my postgraduate degree, and also have an MBA with Henry Business School, University of Reading in the UK. So I'm the Managing Director for HG&H, and we are the supplier of a standardized patented botanical ingredient called Nektium totosum. Prof, you have to forgive me. I know you added cannabis in your outline, but because of my position, I have to speak to what we do supply, which is essentially Nektium. However, the principles do apply. So the idea behind this presentation is to just share some best practice of what we've done in terms of standardization and quality control and quality assurance of our extract using Zembrin as a case study. So just to also reiterate what Prof said, I was quite fascinated that your numbers are significantly different from mine when it comes to market size. But what I did was that I included the food category as well. Because a lot of our products are also, it starts from food and food supplements, plant-based medicines, as well as cosmetics and beverages. So it is quite a significant number, whichever way you look at it, either 70 billion or 170 billion. And this was recorded in 2019. The largest markets that are quite active in these categories, the United States, Korea, Europe, particularly Germany, but it's also cut across Europe. And also there is an emerging trend, but well-established business presence in India. And as well as China. I mean, I didn't even include China there, as well as China. So what about quality? The quality of herbal products is a concern worldwide. Regulators have challenges assessing the safety, quality, and efficacy of these products. A lot of it also has to do with the fact that there has been very lax or lenient registration procedures that are put in place, and also very relaxed labeling requirements across different countries. So one would clearly see that even with the same ingredients, some markets would treat it as a food, some markets would treat it as a food supplement, or even some markets would like to see it as a medicine. And depending on which market you pursue, you have these challenges because the regulatory landscape is very porous and very diverse. But more importantly, it's all about the patient and consumer protection. A lot of consumers who like to, make use of food or dietary supplements usually use the internet. The Google search group is probably their first port of call. But without stringent regulatory requirements in place, they're very unaware with regards to drug herbal interactions, because some of these plant-based medicines are actually medicines. As we know, some of the well-established pharmaceutical products on the market actually found their roots from plants. And so there is the same principles of pharmacology that apply where a drug or a herbal interaction may occur. You could also have the same scenario with a herbal interaction. Then also there's also side effects, because once again, they have activity. Another reason why the challenges with quality is that for most of these products, we do not have well-established monographs that are available as reference documents to cross-check quality standards of these ingredients. Some companies also make medical claims that are not scientifically proven, but may have significant anecdotal evidence from traditional use and folklore stories. Another issue with regulation is the fact that counterfeits, or you could also experience the illegal spiking with prescription ingredients to sort of give the impression that it really works, and it's a big problem when it comes to natural products. Being natural products as well, there's also, over and above just focusing on the active ingredients, there's a need to also look at contaminants, heavy metal testing, fertilizers that are used in the cultivation practices, which would find your way all the way throughout within the supply chain in the finished product. And in most academic experiments, a lot of the discovery that is around natural products really don't go beyond discovery. It requires a lot of funding to sort of take a plant-based ingredient that has some activity and eventually have it developed for use in the commercial setting. So there is a need to standardize. I mean, if the future is to use herbal plant medicines and obviously develop, get to a point where we could use plant-based medicines, on par with pharmaceutical, standardization is critical. So the question is, where do we begin? How do we begin? I have some thoughts around which plant species are under investigation. Too often we see that the focus is just placed on the genus of the plant, but not necessarily the species. How do we translate traditional knowledge to scientific investigation? Because that's where I think the key starts from the traditional knowledge. How do we achieve consistency within the supply chain from plant to product? And how do we ensure that safety and efficacy is maintained? Because these are natural products, and natural products have the difficult challenge of being variable depending on a lot of climatic conditions that we can't generally control. So the question then becomes, how do you ensure that your cultivar, still has the right chemical profile to achieve a desired effect? Is standardization truly achievable with herbal products with all these variabilities? And I'm of the opinion, yes, it is. It can be. So using Zembrin as a case study, so Zembrin is a trademark that we use for Nektium Tutorium. It's a research, developed, manufactured, and distributed plant extract of Nektium Tutorium. Its origin is from South Africa. Traditional knowledge is from the Khoisan tribe, largely found in the Northern Cape, South Africa. Because Nektium tutorium is a protected species by the Department of Environmental Affairs, we were the first company that's HNH to obtain the bioprospecting permit and also entered and signed into a benefit sharing agreement with the SAN Council. And that's the council that looks after the well-being and interests of the Khoisan tribe. So at the end of every year, we do pay royalty payments back to the community with the benefit of using that money to develop the community, because it was from the community that the initial traditional knowledge was obtained. So it's a standardized botanical skeleton ingredient. We use four main alkaloids, which are generally referred to as the Zembrin alkaloids, as the key markers. The mode of action has been identified as a serotonin reuptake inhibitor, as well as a phosphodiesterase 4 inhibition. So where the serotonin is concerned, it helps with stress and anxiety alleviation. And the phosphodiesterase 4 is what is required for the focus and the cognitive effects. Regarding some clinical studies that we've done, we've demonstrated toxicology and safety through preclinical and human studies. Our extract is manufactured at the EU GMP facility in Spain. We've adopted sustainable cultivation because it's part of the requirement for bioprospecting. We're not supposed to cultivate in the wild. And it's also not advisable to cultivate in the wild because there is an unprecedented amount of variability when it comes to species identification, which will also translate to chemical representation each one. We currently have registration status in Canada, the USA, India, South Africa with pending applications. They are patent and trademark protected. Key indications is for use as a mood elevator, stress alleviator, and improves cognition and aids sleep. So the major potential pharmaceutical application of this, it can be used further down the line, investigated for major depressive disorder, and as well as Alzheimer's disease and the management and treatment of Alzheimer's disease. And some very interesting work that is ongoing there. Applications of it in the food and food supplement category is used teas, gums, beverages, supplements, health bars, snacks, sports, nutrition, and performance supplements. And on the right hand of the screen, you can see that is our Skeleton plant that is ready for harvest. We normally grow it every year, typically from December, January, and we harvest in September. So it's time to actually prepare for our harvest at the moment. The Zembrin powder, so we convert it straight from the plant to the powder that you see there. And these are some of the representative products that are available in the markets that I mentioned. When it comes to discovery, where do we start? Yes, this credit must go to my predecessor, Dr. Nigel Karike. So he's a medical doctor who has a very keen interest in ethnobotany. He did the research. He spent some time in the community because he was trying to understand, how the community makes use of it and what is it of benefit is it to them. Because a lot of that preliminary knowledge must be transferred scientifically into downstream into the formulation for commercial use. So he immediately recognized that there was a lot of anecdotal evidence. What he noticed that the Khoisan tribe, when they were about to sort of embark on long journeys without food and water, there was a plant that they chewed. And, and they could go for days on days, feeling calm, very relaxed, without food and water. And what he also realized was that when they were trying to sort of dance around the fire and sing around the fire, there was of the same plant, but there was a different type of the same plant that they chewed. And that is when, upon he did spend significant amount of time with them. On intimate interactions with them, they demonstrated to him that even with the skeleton petroleum plants, there are two types. They've got the mark type and the wild type. So the mark type is the mild type, and the wild type is the one that gives some sort of hallucinogenic effect. And he did the initial studies, and it would be surprising to note that there is distinct chemical differences between the two types of the same plant in terms of the ratio of the alkaloids that are present in. So our focus as a company was to discover and propagate the MAC type, which is the mild type that gives you the stress alleviation, the keeps you calm, and also keeps you focused. So what we did was that we need to get a permit from Cape Plants Natural, and you locate some wild growing plants, which we used for initial research and to gather information. And once we sought the mother plant, that was what we propagated for under a sustainable cultivation protocol for use. From interviewing the folklore, it was determined that about 1 to 2 grams of plant was chewed when needed. And also very importantly was that the part of the plant that was critical for activity was the aerial part. And there was a general understanding within the folklore that the roots were forbidden. So they were not supposed to chew the roots. Some of the anecdotal evidence here was that these plants were also given to babies, to help with colic and to also help with feeding, and also to help them sleep when they were going through tough times. Yeah. So in our definition, from with my interaction with Dr. Nigel Karike, this is where quality control actually begins. Because if you don't get this part of the research right, you could go off in a tangent, just reading somewhere that Skeleton has some properties and you just want to propagate it for the benefit of commercial gain without understanding the science. So what we did was that we tried as much as possible to replicate the quality control and the scientific research based on the anecdotal evidence that was collected. And this was also, so when Nigel started working on it, it was true. I mean, if you look at the different types of Scilitium, just by the donation of the leaves, there were some differences that were quite obvious. That the Imaricidium type of Scilitium totosum usually had a narrow leaf donation profile, while the totosum type had a broader leaf donation, which also gave to show that if you don't have this is not so obvious to the average person. You really need a taxonomist to be able to tell you that identify the different types of plants just by looking at it. So Scilitium, there are about 8 species of Scilitium that is available to us, or that is known to us, and only 2 of those species have some sort of chemical activity. And how do we do this? So we normally identify those through DNA. So you want to be sure that you've got the exact right species to start with. Otherwise, if you cultivate the wrong species type, you will have no activity. And I think I have a slide that speaks to that later down the line. So the chemistry, four key chemical components is what we track or we use as markers to attest the chemistry of our extract. So these are the mesembrinol, mesembrinone, mesembrin, and mesembranol. What we've identified is that the mesembrin component is and that's the one here, is what is responsible for the hallucinogenic effect. So when we identified and tested analytically a wide range of products on the market that claim to have Scilitium totosum, you'll be quite surprised to see the variation amount of mesembrin in those products. So we, as part of our quality control measure, we ensure that our mesembrin content does not exceed it's less than 5%, and that's the one you see over there. Okay, and our total component of mesembrinol and mesembrinol can exceed 80%. So this is just an MS chromatogram of our extract. Reference standards, we manufacture our own reference standards from our dry plant material because we need reference standards to be able to qualify and identify these alkaloids in the first place. So reference standards is important. Without reference standards, you will not be able to adequately quantify these things. And the reference standards is also quite an expensive exercise to have, but it's necessary. So this slide speaks to the chemotype variability. I mentioned earlier that there's about 8 known species of Scilitium available. And as you can see, the Imaricidium species has no chemical activity here, and the Archery species also has no chemical activity, and Variance also has no chemical activity. The only 2 species that have the presence of alkaloids are the totosum species and the quasicula species. This is a research that we embarked on and it's still ongoing, but this definitely tells, very quickly that if you do not understand and control the type of species that you're working with, you could potentially be growing a Scilitium and Imaricidium and growing that in large scale and basically selling placebo in the market. Which would not be, useful to the end user in general. Or you could be growing a species that has a very high mesembrin content and putting that on the market, which would have hallucinogenic effects and side effects. So there's a big need for regulators to have a firm control on some of our plant-based sources, so that we control the effect of these products on patients at the end of the day, and make sure that patients are not being ripped off when they purchase herbal products. So the DNA is very important. So this slide speaks to some 8 commercial products that were ascertained. This was not a study by us. And as you can see, the study speaks to looking at the variability of the 4 key alkaloids in 8 commercial products. And you can already see that the missing green, which is the green one, you can see the variation of it. Commercial product number 4 and number 8, and as well as 1 and number 3 will be of major concern. I mean, if end users take those commercial products, they would definitely experience hallucinogenic side effects. The ideal commercial product that we would want to mimic is number 5, where the mesembrane content is about less than 5%. And even in that scenario, it's a little bit more, but it's the best one of all these. We've also just concluded our own in-house analysis of about 72 products that we purchased globally. And it's amazing how the variability of those products really look. There is definitely no standardization when it comes to these products on the market. And it's a bit of a concern, it really is. So just to show you what we're doing from our side, from a cultivation perspective. It took us quite a while to achieve it, but on this side here, that is our nursery where we start off with the nursery, the seedlings, and they start off in the nursery, and after 3 or 4 months, they are transferred into the shade nettings where they grow to maturity. So at this side as well, my team is sort of inspecting the quality of the seedlings with the farm manager. And it also has to be a farm that practices good agricultural practices. We maintain documentation, we track everything and everything. You know, the soil material that is used, the peat material that is used. Shingi sort of keeps track of all the NSDS, the chemical composition. We keep track of the amount of rainfall or drought. About 2 years ago, we experienced the drought. And the effect of drought can be quite pronounced because when there is no water in the dam, because obviously all these active cultivation is the irrigation is from the dam. You know, you increase the amount of heavy metals and other chemicals that are present, which will eventually find their way into the plants and will eventually find their way into the finished formulation, which will not pass quality. So there's a lot of things that need to be controlled, you know, and we have to work with partners that adopt good agricultural practices and keep relevant documentation to be able to ascertain that. So then once harvest is reached, which is typically in the month of September, we start cultivating, we cut it out. So remember when I was giving you the feedback on the folklore, so we don't touch the roots. So we cultivate, we just cut out the aerial parts, we roll them back into bags and pack them in trays, and we dry them. So here you can see the drying preparation going on, and the wet plant material is put into the ovens, and the ovens are set at specific temperatures. The key parameter for us here is moisture. So we try and achieve a moisture content of just about 5 or less than 5%. And we do that by regularly checking the moisture analyzer. And then we sort out food drying, and they're put into the bags. And after that, it goes out for steam sterilization before we put it to storage. So this is just an example of what the dry plant material looks like. And on here, there's a certificate of analysis that is issued at this level, which sort of confirms the alkaloid presence, you know, and the mesembrane. So we keep an eye on the mesembrane content just to keep it, make sure that it's less than 5. We keep an eye on the moisture content, and we do a whole suite of heavy metal testing and as well as microbiology tests post steam sterilization to make sure that it complies. So these are some of the tests that happen, and we have to maintain it throughout the entire supply chain. On here, once we are ready to make the extract, we ship so first we have to apply for a phytosanitary certificate to be able to export out of South Africa to the EU, and in the EU, we send it to EU GMP facility where that converts it from the dry plant material into an extract. It's a typical ethanolic extraction procedure. The reason why we chose that method was because also from the folklore, that is how they extracted it. Or when they wanted to use it, they would put it into a bit of alcohol and they will extract it. Sometimes used to drink it and give it to children. So it was important that we chose the exact same extraction procedure. There's a separation just to get rid of some of the solid waste. And after that, the ethanol is evaporated, it's spray dried, and we ensure a standardization of the total alcohol content to zero point 0.4 weight per weight using a maltodextrin carrier. So that's the second aspect of the standardization. The first aspect is keeping an eye on the presence of the alkaloids, but more importantly on the ratio of the alkaloids. And then the second aspect of the standardization is that we ensure that we have 0.4, not more than 0.4% weight to weight once the extract has been made. We do many tests, which I'll show you on the next slide before release is done. So this is just a picture of what I think is the 1 kilo bag of our extract. And it also gets issued with a C of A. Also reconfirming the DNA status just to make sure that nothing has changed. We reconfirm the fingerprint once again just to make sure that in the extraction process nothing has changed. We measure the maltodextrin content. We normally use silicon dioxide just for flowability, and we keep an indication of the plant extract ratio, which is typically 2 to 1 or 2 parts to 1. And the quality team pretty much just goes through all these parameters, making sure that all the physicochemical properties and the solubility as well as density is all within spec. And also, once again, the big thing here is ensuring that the total alcohol content is 0.4, and as well as the ratio of the four alkaloids are within spec with the Zembrin and the 5. And also keeping an eye on the moisture content. We have done stability, so our current stability on the extract 3 years. We did it at zone 2, climatic zone 2. We're redoing the stability to see if we can get an extension to 5 years, because a lot of our older extracts seem stable. So just to give you an overview of the types of tests and the number of tests that we do to get to the extract stage, right from cultivation. You can clearly see that there's a lot of testing points that we do, from phytochemical analysis, DNA plant analysis, pesticides and contaminants, right at the cultivation stage. When we dry, the key parameter there is moisture content analysis and the phytochemical analysis. Before steam sterilization, heavy metals, pesticides, microbial analysis, and phytochemical. Labeling controls, temperature controls, because we ship, we normally store under cold storage, and we ship under refrigerated conditions from 2 to 8 degrees to Las Palmas. And once it gets there, a lot of these tests need to be retested just to make sure that it meets the EU specification. So this is also another thing. If you look at the specifications of the same ingredient from South Africa and the EU, our specification is very light. But because we're shipping it to an EU GMP plant facility, it's important that we have to match the specification for that market with the assurance that we're not introducing other things into it. We also do an allergen screen just to make sure that there is the absence of any potential allergens, which would have probably come from contamination. And once the extract is manufactured, all these tests needs to be redone, right from DNA, allergen, microbiology, heavy metals, pesticides, solubility, density, residual solvents, just to make sure that the ethanol is out of there, particle size, and as well as nutritional analysis. So the testing that is required is quite heavy. And these tests are necessary because this is what gives you the confidence that when you release the product and when the product becomes available for sale, you've got the confidence to know that you are you've cultivated the right species, and you've cultivated the right chemistry, and as well as there's the absence of unwanted chemicals and pesticides and contaminants that you don't want to have in the product. So in conclusion, standardization is achievable, but it will not be achievable if the ethnobotanical research is poorly executed. And I think that is the main part that a lot of companies don't focus on. They just try and grab their hands on a plant, and because of what they've read about the plant, and they just want to grow it, and without testing the quality. And another thing that we've also realized is that some of the other companies like to benchmark their cultivar against the clinical research and quality work that they've done, giving the false impression that because it's skeleton, it's all the same, which is not technically true. There's a need for DNA sequencing to make sure that the right species is being done. Analytical method development, validation is critical. Without reference standards, we will not be able to first identify and quantitate. Cultivation process needs to be controlled, and as well as needs to be sustainable. Where possible, just avoid wild cultivation, because if you just introduce a very high amount of irregularity, regular quality control testing is necessary. Key steps within the supply chain, and very important, the extraction technique should always mimic the traditional knowledge. But unfortunately, all this requires significant investments, which is what deters most companies from achieving it and getting it. And these are some of the references that we use, and many thanks to my colleagues for helping. Okay, Prof, over to you.
Thank you very much. Yeah, very interesting. I think you got a lot of people excited there. So there are some questions in the chat box, and you can respond to those as we go into the next presentation. If we have time at the end, we can also just come back to that. Okay, but thank you very much. We will now go to Aika.